Welcome to the deep dive. Today we're going to be looking at a world that most people don't even realize exists.
Yeah.
It's the fascinating realm of analytical chemistry. Specifically, how scientists can actually separate and study
Right.
individual molecules.
Like the teeny tiny little pieces of stuff.
Yeah, and this isn't just lab coat stuff, by the way.
Oh, no.
This science is behind everyday things like, medicines,
Yeah.
crime scene investigations, even understanding the air we breathe.
Absolutely.
And we've got some seriously cool techniques to cover today from your source material.
Yeah.
Gas chromatography, ion mobility spectrometry,
Right.
and some variations that'll make you go, huh, that's clever.
It's like having a molecular magnifying glass.
Yeah.
Letting us see the building blocks of matter in a whole new way.
All right, let's start with something I find super intriguing.
Yeah.
Gas chromatography,
Okay.
or GC.
Uh-huh.
The source material describes it like a microscopic racetrack for molecules. What's the deal with this tiny race?
So imagine a track where molecules are competing based on how volatile they are,
Okay.
how easily they turn into a gas, and also how they interact with the track itself.
Okay, so some molecules are kind of...
Yeah, so it is a bit like those Ninja Warrior courses where some contestants fly through and others get stuck on the obstacle.
Oh, yeah, I can see that.
Yeah.
Okay, so some molecules zoom ahead while others lag behind.
Exactly.
Makes sense.
Yeah.
But how does this translate into actually analyzing stuff?
Well, so GC is really good for analyzing mixtures that have volatile components,
Okay.
lightweight components, and things that are thermally stable.
Okay.
So things like essential oils or the compounds in a fragrance.
So breaking down a perfume into its individual scents.
Exactly.
That sort of thing.
Pretty cool.
Yeah.
The source material also mentioned that GC has a few aliases.
It does.
Vapor phase chromatography.
Yeah.
And gas-liquid partition chromatography.
Right.
Why so many names for the same technique? Well, each name highlights a slightly different aspect of the process.
Okay.
So vapor phase emphasizes that the separation happens between a gas and a liquid.
Gotcha.
And gas-liquid partition kind of dives into how the molecules are constantly interacting with both phases.
Okay. Yeah, I can see how each name gives a slightly different perspective.
Yeah.
And the source material was pretty clear that GC and liquid chromatography are distinct, right?
Absolutely.
Okay.
They share some similarities, but there are three key differences that set them apart.
Okay, cool. What's the first one?
So the first one is the separation mechanism.
Okay.
So in GC, it's all about the interaction between a moving gas
Okay.
and a stationary liquid.
Okay.
Whereas LC uses a liquid mobile phase.
So different playing fields for the molecules to move through.
Precisely.
Got it. What are the other key differences?
So the second is in GC, scientists can very precisely control the temperature of the gas.
Okay.
And that directly affects how the molecules behave.
And the third one?
And then third, a compound's concentration in the gas phase. Okay.
Depends solely on its vapor pressure.
Vapor pressure.
Yeah.
Isn't that like a measure of how easily a liquid evaporates?
Exactly. You got it.
Okay.
It's like that pot of boiling water we were talking about earlier.
Yeah.
Molecules with lower boiling points or higher vapor pressure
Okay.
escape as steam first, just like in GC.
Oh, okay.
They're the speed demons on our molecular racetrack.
Okay, that's a great analogy. I can really picture it now. So we've got our molecular racetrack.
Yeah.
But what about the actual hardware?
Right.
What makes up a GC system?
Well, first you need a carrier gas to carry the molecules along the track.
Okay.
It needs to be chemically inert so it doesn't react with the sample, and highly pure.
Okay.
And so usually scientists will use things like nitrogen,
Okay.
helium-argon, or carbon dioxide.
So the carrier gas is like the wind propelling our molecular athletes.
Exactly.
What happens next?
Then you've got to get the sample into the system.
Okay.
That's where sample injection comes in. Okay.
There are a few different ways to do this, each with its own pros and cons.
Okay.
But we don't need to get bogged down in those technicalities.
Fair enough. But the source material did mention something about columns.
Yeah.
What role do they play in this molecular marathon?
The columns are the heart of the separation process.
Okay.
They contain that stationary liquid phase, our metaphorical racetrack, which interacts with the molecules and influences their journey.
Okay.
There are packed columns, which are like a crowded obstacle course, and capillary columns, which offer a smoother, more defined path.
And the source material specifically mentioned WCOT, SCOT, and FSWC columns.
Yeah.
Those sound pretty technical.
They are indeed.
What's the difference between those types?
So each type has a slightly different internal structure that affects its performance.
Okay.
Kind of like how the material of a running track can influence a runner's speed.
Okay, yeah, I can see that.
But essentially, they all provide that long, thin pathway for the molecules to separate.
Okay, I can see how getting too deep into those specifics might lose some listeners.
Yeah.
What about the column oven?
Right.
I imagine temperature control is pretty crucial in this delicate process.
Absolutely.
Okay.
The column oven keeps a very precise temperature throughout the separation process.
Okay.
It can be a constant temperature called isothermal,
Okay.
or it can gradually increase, which is known as temperature programming.
Speaking of temperature, there was a visual in the source material that really caught my eye.
Oh, yeah.
It showed how changing the temperature can actually affect the shape of the peaks
Uh-huh.
in the chromatogram.
Yeah.
The output we get from the GC.
Right.
Why is that shape so important?
Well, that shape tells a story.
Okay.
Sharper peaks mean better separation,
Okay.
while broad or overlapping peaks suggest that the molecules aren't separating effectively.
Okay.
It's like looking at the finish line of a race. Yeah. Well-defined gaps between runners mean a clear winner, while a jumbled mess makes it harder to tell who came out on top.
Yeah, that makes sense. So a clean separation is like a photo finish,
Exactly.
giving us a clear picture of each component in the mixture.
Right.
Fascinating.
Yeah.
Now, before we move on to ion mobility spectrometry, I'm curious about two specialized types of GC that were mentioned in the source material.
Okay.
Pyrolysis GC
Yes.
and comprehensive two-dimensional gas chromatography.
All right.
What can you tell me about those?
Okay, so pyrolysis GC is like a super-powered version of GC that tackles solid samples.
Okay.
Imagine heating a solid to insanely high temperatures,
Right.
breaking it down into gases that can then be analyzed using traditional GC.
Oh, wow.
It's like blasting a rock into tiny pieces to study its individual components.
Oh, wow. That's intense.
Yeah.
And what about comprehensive two-dimensional gas chromatography?
So think of it like going from a single racetrack to a whole network of interconnected tracks.
Okay.
So GCSGC uses two columns. Okay.
With different separation properties, creating a two-dimensional separation space.
Gotcha.
It's like having a preliminary race.
Yeah?
Followed by a final, super precise race.
Oh, wow.
Giving scientists incredible resolution for complex mixtures.
Okay, so it's like taking our molecular racetrack.
Yeah.
And adding a whole new level of complexity and precision.
Exactly.
That's mind-blowing.
It is pretty cool.
I think we've covered a lot of ground with gas chromatography.
Yeah.
But get ready for a whole new adventure, because next up, we're taking our molecules to the airport with ion mobility spectrometry.
This is where things get really interesting.
Okay.
We're not just separating molecules based on their speed and affinity for a stationary phase anymore. We're analyzing how they fly.
All right, bring it on.
Okay.
Okay, so we've left the racetrack, and now we're at the airport with ion mobility spectrometry, or IMS, instead of speed and agility on a track.
We're talking about how molecules fly through an electric field.
Yeah.
I'm picturing something like a microscopic wind tunnel.
That's a great visual.
Am I close?
Yeah, so in IMS, we ionize the molecules, give them a little electric charge, and then send them through a drift tube under the influence of an electric field.
Okay.
Just like different airplanes have different aerodynamic profiles, these ions move through the drift tube at different speeds depending on their size, their shape, and their charge.
So it's not just about how fast they travel.
Right.
But also how they navigate this electric field.
Exactly.
That makes sense.
Yeah.
But the source material mentions something called collision cross-section.
Yeah.
Or CCS. Is that like measuring the wingspan of our molecular airplanes?
You're on the right track.
Okay.
CCS is basically a measure of how much space an ion occupies.
Okay.
As it bumps and jostles with other gas molecules in the drift tube. It's like a molecular fingerprint that reflects the ion's size and shape. And it's particularly useful for distinguishing isomers, molecules with the same mass but different structures.
Oh, so it's like those molecular twins we were talking about earlier.
Exactly.
They might weigh the same,
right? But
their CCS can reveal those subtle structural differences.
Yeah.
It's pretty neat.
It is cool.
But the source material also threw out a whole bunch of IMS acronyms.
It did.
DTIMS, TWIMS. TIMS, FAIMS, DMA.
Yeah, that's a mouthful.
Are these like different terminals in our molecular airport?
That's a fantastic analogy.
Okay.
Each platform represents a slightly different approach to sorting ions based on their mobility.
Right.
So DTIMS,
Mhm.
or drift tube ion mobility spectrometry,
Yeah.
is the classic approach.
Okay.
It's like a straight runway where ions drift under a constant electric field.
Okay, so that's our basic takeoff.
Right, very straightforward.
What about the others?
Okay.
How do they spice things up?
Well, TWIMS, or traveling wave ion mobility spectrometry, adds some flair. Instead of a constant field, it uses a series of voltage pulses
Okay.
that act like waves propelling the ions forward in a more controlled way.
So it's like a moving walkway that guides the ions along.
Exactly.
Interesting. So it's a bit more dynamic than the simple drift tube.
It is.
What about TIMS?
Okay, so TIMS, or tracked ion mobility spectrometry, is like a molecular holding pattern.
Okay.
It uses a combination of electric fields and gas flow
Okay.
to trap ions in a specific region of the drift tube,
Ah.
allowing for very precise measurements of their mobility.
Gotcha.
It's like having a security checkpoint where the ions have to pause for a detailed inspection.
Okay, so it's all about getting a really clear picture of each ion's flight characteristic.
Precisely.
What about FAIMS and DMA?
Okay, so FAIMS and DMA are like specialized filters.
Okay.
They use changing electric fields to selectively transmit ions based on their mobility.
Okay.
It's like having different gates at the airport, allowing only certain types of aircraft through.
So it's all about choosing the right tool for the job depending on what you want to analyze.
Exactly.
But I'm curious,
Yeah.
why is all this separating and sorting so important?
Right.
What can we actually do with this information
Yeah.
once we've identified our molecules?
That's the key question, isn't it?
Yeah.
IMS, especially when combined with mass spectrometry or IMS-MS, opens up a whole world of applications.
Okay, let's dive into those applications.
Yeah.
What are some of the most exciting things we can do with IMS-MS?
Well, as we mentioned earlier, IMS is a game changer for isomer separation.
Mm-hmm.
Being able to distinguish between those molecular twins that have the same mass but different structures
Right.
is crucial in fields like drug development, food science, and environmental monitoring.
The source material mentioned the example of separating diglyceride isomers
Yeah.
based on their double bond orientation or chain length.
Exactly. Those subtle structural differences can have a huge impact on a molecule's biological activity
Okay.
or its function in a particular process. For example, one isomer might be a beneficial nutrient, while another could be a harmful contaminant.
So IMS-MS is like a molecular detective helping us untangle those subtle clues
Yeah.
that can make all the difference.
It is.
What other mysteries can this powerful technique solve?
Another key application is signal filtering.
Okay.
Imagine you're trying to listen to a specific conversation in a crowded room.
Yeah.
It's hard to pick out the signal you're interested in amidst all the noise.
Right.
IMS-MS helps clean up mass spectra by filtering out background ions
Okay.
and focusing on specific molecules of interest.
It's like having noise-canceling headphones for your mass spectrometer.
Exactly.
You can actually zero in on the molecular conversations that matter.
Yeah.
That's pretty impressive.
It is a powerful tool.
Anything else?
Absolutely. IMS-MS is becoming increasingly important for identifying unknown compounds.
Okay, how so?
Remember those collision cross-sections we talked about?
Yeah.
Well, scientists are building extensive databases of CCS values for a wide range of molecules.
So it's like having a fingerprint database for molecules.
Exactly. By comparing the CCS of an unknown compound to those in the database, researchers can get valuable clues about its identity.
Oh, wow.
It's a powerful tool for discovering new molecules
Okay.
and understanding the composition of complex mixtures.
Wow. It sounds like IMS-MS is really pushing the boundaries of what's possible in analytical chemistry.
It is a rapidly growing field.
But are there any challenges or limitations to these techniques?
Of course, no technology is perfect.
Right.
One of the biggest challenges is the need for standardized methods and comprehensive databases.
Okay.
The field of IMS-MS is still evolving,
Yeah.
and there's a lot of work to be done in terms of creating consistent protocols and ensuring the accuracy and completeness of CCS databases.
So it's like having a library with some missing books and a few mislabeled shelves.
Yeah, that's a good way to put it.
There's a ton of potential, but we still need to organize and refine the information to make the most of it.
Exactly, but the future of IMS-MS is incredibly bright, with exciting possibilities for developing new applications and improving our understanding of complex biological systems.
Yeah.
From personalized medicine to environmental monitoring to materials science, the potential is truly vast.
This has been an incredible journey so far.
It has.
From the molecular racetrack of GC to the molecular airport of IMS.
Yeah, a whirlwind tour of the molecular world.
We've seen how these powerful techniques can separate and identify molecules with incredible precision.
Right, with amazing accuracy.
But what does this all mean for the listener?
That's the important question, right?
Why should they care about these seemingly complex scientific tools?
Well, let me tell you.
Well, we've explored the ins and outs of gas chromatography and ion mobility spectrometry.
Yeah.
We've seen how scientists can separate and identify these molecules with incredible precision. But I think it's time to answer the big question.
Okay.
Why should anyone besides a chemist care about this stuff?
Well, that's a fair point. It might seem like this is all happening in some far-off lab.
Right.
But the truth is, these techniques have a huge impact on our everyday lives.
All right, I'm all ears.
Eh?
Give me some real-world examples.
Yeah.
How does this molecular detective work actually translate into, like, tangible benefits?
Okay, well, let's start with the pharmaceutical industry.
Go ahead.
Separating molecules is crucial for drug development.
Okay.
It ensures that medicines are pure and effective. It's like making sure you're getting the right ingredients in a recipe.
Yeah.
You don't want any unwanted surprises.
Right, so it's about making sure you get the right medicine at the right dose.
Exactly.
Without any contaminants that could be harmful.
Precisely, and it goes beyond just creating new drugs.
Okay.
These techniques are also essential for understanding how medications are broken down and processed by the body.
Okay.
Which is vital for personalized medicine.
That makes sense, figuring out the optimal dosage and treatment plan for each individual.
Right.
It's like tailoring a suit one size doesn't fit all.
Perfect analogy.
Okay.
Now let's move on to food science.
All right.
Separating molecules helps us analyze food composition,
Okay.
identify potential allergens, and even detect contaminants or adulterants.
So it's about ensuring the safety and quality of the food we eat.
Yeah.
From farm to table. It's like having a molecular watchdog, making sure our food is what it claims to be.
Precisely. And then there's environmental monitoring.
Okay.
These techniques are used to analyze air and water samples, identifying pollutants and tracking their spread.
So it's about safeguarding our planet, understanding the impact of human activity, and finding ways to protect our environment.
Absolutely.
Pretty important stuff.
Essential.
I'm starting to see how understanding molecules at this level is really about understanding our world, its complexities, and the interconnectedness of everything.
Yeah, like having a new lens to view the world through.
It really is.
Yeah.
And it's not just about understanding.
Right.
It's about problem-solving innovation and creating a better future for all.
Absolutely.
This has been an incredible deep dive into the world of separating molecules.
It has.
We journeyed from the microscopic racetrack of gas chromatography to the molecular airport of ion mobility spectrometry.
A whirlwind tour.
And we've seen how these seemingly complex scientific tools have a profound impact on our lives.
They really do touch every aspect of our world.
It feels like we've only just scratched the surface of what's possible.
Oh, absolutely. There's so much more to explore.
So to our listener, we leave you with this.
Okay.
Now that you've glimpsed the power of separating molecules, what new possibilities do you see? What mysteries could be solved? What breakthroughs could be made using these incredible tools?
It's an exciting time to be in this field.
Until next time, keep exploring, keep questioning, and keep diving deep.